want any student who has the drive, who has the passion, we want to give them the opportunity to succeed. So get their foot in the door, that first step, which is a job, but we are also looking at really getting them ready to succeed in the career. And we are doing this all through the internship part. So we're really rethinking internship as a way to expand opportunities and make the STEM force more inclusive. And I'm an optimist, a diehard optimist. I believe in the STEM community. I deeply believe in students. So looking at the summer numbers, it's no longer just optimism. We have numbers which prove that we are going towards these goals. We can make STEM more inclusive and really give opportunities to students. And I'm also an engineer, so I was at Intel doing something totally different. The reason I left Intel to start STEM Away was I had this idea about how we can achieve it, and that's where the mentor chains comes in. So the concept of mentor chain is, it's literally like a chain of mentors. So we have the industry experts in bioinformatics. It was a postdoctoral fellow, Ali, who is helping the leads. And the presentation that you're going to be seeing today is from all the leads. So the leads are taking the learning from Ali, and then they're teaching, helping their teams learn bioinformatics in this particular pathway. And it has been challenging. Our whole goal is to really push the students on every front. This was particularly so for bioinformatics because everybody came in pretty much new to the area. So what they've achieved is, what you see they've achieved is basically starting from almost zero. So there have been lots of challenges, lots of frustration, but what has been incredible and what has been really fulfilling is the achievements at the end of it. So I'll just hand it over to, you can listen to it from the student leads themselves.
Thank you, Devalina. So what is very unique about the STEM Away platform and setup of the internship is that the project goals are set by the mentors and leads themselves, making it really unique to each pathway. The program is comprised of real-life projects selected to develop skills for specific career pathways, and in this case, bioinformatics. So the teams were constructed to be at its core collaborative and team-centric, which was especially essential within our all-virtual platform. Being completely virtual was a new challenge for all of us, as all of the traditional aspects of forming a productive and high-performing team had to be adapted to our new environment. Also, as leads, one of our biggest goals was to create a safe environment for all of our teams that allowed participants to freely and without hesitation ask questions, push their creativity, and expand the boundaries of the goals, of the project goals that we set out. We really had to come up with non-conventional ways to build team camaraderie and form relationships. Effectively, we came up with engaging happy hour events, had icebreakers before all of our early meetings, and always found ways to increase networking between teams. So one of these happy hour events that I'm personally quite proud of myself was the paint night, where all of our participants followed a YouTube painting tutorial and each painted their own version, as you can see here. Additionally, through STEM Away, self-reflection was a skill that we really wanted to enhance. With periodic personal self-assessments, we encourage participants to assess their learning and progress and set new weekly goals. We wanted to instill this habit into our teams, as being able to track growth and improvement through self-reflection is a very important skill to possess. Compounded with that, each week we asked our teams to give us feedback on the way they thought the project was going and how they wanted to see improvements in us as leads and in the project. We wanted to constantly find ways to improve ourselves as leaders and proactively tackle any problem that the teams face both internally and externally. So for example, one of the suggestions we had was to give more information on resume building. The following week, I put together an informative presentation that gave insight onto all the sections to include in a resume, what to highlight or avoid, and how to go about applying for jobs. Furthermore, we were asked to provide further instructions for the deliverables to help accommodate those who were not as experienced with R. As a result, we went back to the mentors and as a whole really focused on making them clearer and easier to follow. Essentially, the bioinformatics pathway was driven by hands-on learning. Leads and mentors gave participants the necessary tools and access to information to learn independently and at their own pace, while still abiding by the internal team set goals. We did so by stressing communication, organizational, and troubleshooting skills to empower participants to take their own initiative to learn, creatively problem solve, and complete the tasks at hand. The final result was end-to-end execution involving both lateral and strategic thinking. Through the bioinformatics pathway, we wanted our participants to first learn the skills and then the application. So what that means is that we wanted to give them the necessary tools and skills to learn the overall pipeline and then be able to apply it to any scientific paper in order to reproduce the results. We had 3 main steps in the overall pipeline, as you can see here: normalization and quality control, DGE analysis, and finally functional analysis. More details will be covered in the following slides, as each lead will talk about a specific section of the pipeline, which will set the stage nicely for our panel discussion at the end of the presentation. Also, during the discussions, we welcome anyone to join in with questions or comments. Once participants had a fundamental understanding of the overall pipeline, we asked teams to apply it to the following research paper and reproduce selected results. Just to give a brief background on the paper, this study analyzed microarray data to explore potential prognostic markers in colorectal cancer via the construction and in-depth analysis of a competing endogenous RNA network. The project as a whole was definitely a process where the leads were also learning in parallel with the participants. At the beginning especially, we were adjusting the scope of the project and the specific deliverables we wanted our teams to achieve in relation with the pipeline. As we learned more about the experience of our participants, what they wanted to achieve from this internship and how we could provide that, we were continuously adapting and improving the project. For example, we initially chose a different scientific paper than the one presented in front of you, but we then decided to switch to this one as our participants noted that they wanted to see a significant biological connection to support their analysis. It was really great to see how flexible all the leads were by adapting and overcoming the hurdles that were presented to us. I would now like to introduce the other leads for the June and July sessions, Yves, Annie, Alicia, and Anya, who will go on to talk more about the specifics of the pipeline and the Bioinformatics Project. Additionally, I would like to introduce Isha, who is our project manager for both teams, as she will be discussing the project management side of this pathway.
In project management, we were tasked with managing remote teams of 10 to 30 interns spread all over different time zones and across the world. In the July session, we had around 10 students per team, which is a very optimal and manageable number, but we had much larger numbers in the June team. Initially for the June session, the plan was to make 4 smaller teams, but we decided to restructure around 2 weeks into the project and change to 2 large groups to better utilize the experience of the leads. We also had to come up with project specifications for what we were to develop, and we had to develop those and establish the scope of the project in order to make a successful end product. In the June session, we started with goals and deliverables on the fly, but in the July session, we came up with a very clear timeline in advance of the session. As you can see from the timeline, we spent a while on quality control. In the June session, we received some feedback that we ramped up too quickly, so we elected to switch quality control to 2 weeks to allow for a more gradual buildup. We also spent a while on functional analysis in weeks 5 and 6 due to the level of difficulty. And now we have Yves talking about the quality control step.
We know microarray experiments produce a set of images which are transformed into numerical values representing intensities. As in any statistical analysis, and particularly in image analysis, the quality of the data must be checked first. Interestingly, there was not much information about quality control in the paper we chose. The goal of quality control is to determine if the whole process to generate our data has worked well enough so that the data can be considered reliable. Moreover, we perform quality control to identify the outliers in our dataset. The procedures of generating microarray data is complex. Outliers can be experimental artifacts or just genuine consequences of biological diversity. However, an outlier sample caused by biological diversity may be different from other outliers, possibly harder to detect and questionable if it is an outlier or not. Another reason why we decided to do quality control is to increase the signal to noise ratio, which can be expressed as the amount of biological signal relative to the amount of noise. It is important to mention that there is not a standard or protocol to perform quality control. So we decided to give the participants just a set of packages that they can be used to do quality control. And the last thing about quality control I want to add is that we can divide this step into this quality control, this whole step into two different categories. We have the intra-sample quality control that is used to assess the quality of expression level of a particular spot and to control the uniformity between probes of the same array. While the second subcategory is experimental level or inter-sample quality control that is used to compare all the arrays in the experiments to identify outliers and batch effect. After this brief introduction to quality control, I would like to share with you the findings of our quality control step. As you can see, we use different packages. That is because each of them give us different information about our datasets, and also to show the participants that quality control can be done in different ways. Since we have a really diverse group of students and most of them without any experience in art and bioinformatics, we wanted to give as many tools as possible, of course, taking into account the diverse background of the group so that they could apply them in autonomous way after the internship. The vast majority of the packages we use are from Bioconductor. Some of the packages are, for example, simple FE, array quality metrics, FAQC report, and FAPLM, as you can see in the two graphs in the scheme. The central focus of FAPLM is implementing methods for fitting probe levels and methods and tools using this method. The left plots show the result of relative log expression, while the right plot shows the results from the normalized on-scale standard error. And as you can see, we have that, how can we detect outliers? It's pretty simple to detect outliers. By looking at the graph, we can see that those samples that are far away from the overall mean, then we can identify them as outliers. And then another method to identify these outliers is also to look at the interquartile range. As you can see, those samples having a big interquartile range and pretty far from the overall median we consider those as outliers. So from these two graphs, we can say that for us, the outliers are sample 1, 11, and 34. The next step in quality control and data processing was to normalize our data and perform background correction. A background adjustment must be performed to remove signal due to non-specific hybridization, that is signal emitted by other things than sample hybridized to probe, while normalization of data must be done to correct systematic biases due to causes such as different dye assumption, spatial heterogeneity in the chip or other stuff. As we know, performing a microarray experiment is pretty difficult, so we can have this kind of errors. Then, what do you see here? What you see here are just two PCA plots. On the left, you see the PCA plot of the unnormalized data, and on the right, you see the PCA plot of the normalized data. We do this to show the participants that normalizing data is pretty important, because from the first plot, we could say that sample 11 is an outlier just because it is outside from the ellipse, including all the samples in this cluster. And we will also say sample 21 and 26 are outliers. But after normalization, what we realize is that sample 11 and 34 are the real outliers here. But also sample 1 is, and we can consider this an outliers thanks to the analysis done before, for example, as I showed before, using RLE or unused. And in this internship, we are leading our teams through these complex steps. For that, we need a solid project management structure.
As Eve just mentioned, we use project management tools to work on the project as efficiently as possible. Our main method of communication with the interns was through the STEMaway forums. G Suite was also very important for us. We used Google Calendar extensively to schedule our meetings. All the leads would meet on Sunday afternoon and plan out the meetings for the week. This ensured that everything was posted and ready to go at the start of the week. Deliverables, meeting recordings, and notes were posted on Google Drive. And we used Slack for emergency communications in case the meeting link was broken or for quick reminders to turn in deliverables. We used Drive as leads to do the weekly surveys Erin had mentioned earlier. These surveys help gather feedback on the intern's experience. The overwhelming majority of the feedback on these surveys was positive. However, there were a few negative responses as well. This was quite a shock for us since it was our first time dealing with negative feedback. We were putting in a lot of work and weren't sure what else we could do as leads. So we had to talk with Ablita, who made us feel better and showed us to focus more on the positive and take negative feedback as constructive criticism. We also introduced GitHub because we needed a way for students to submit their codes. However, it proved to be an additional complexity that some students were struggling with, so we made it optional. Students could still submit code via the Google Drive as they had done before, but at the same time, they got introduced to the basics of GitHub, which is an essential tool for future data scientists. With this project management setup, we were in great shape to move on to the DGE analysis.
Awesome, thank you, Isha. So the next step on the pipeline was the differential gene expression analysis. And so the differentially expressed genes were ones that had significant difference in read counts between the different conditions, in this case, cancer or non-cancer. And so the main question we had was, what was the best way to standardize this analysis and which tools could we use? So we talked extensively to the mentors who had kind of come up with a good pipeline for us, and they had told us to use the lemma package. And so that proved to be a really great way to do the DGE analysis. One of the biggest challenges here, though, was that this was kind of a really big step up from the previous packages they had used because it was so extensive. And so one of the ways that we kind of combated that was by having more frequent office hours and actually inviting the mentors to show up to those as well, so they could give more of a deep insight into the packages and what their use was. And so here you can just kind of see the three main steps we took for that week of the DGE analysis. First was the annotation step, which was conducted on the data to match the probe IDs to the gene symbols. Next was gene filtration, which was used to remove non-relevant genes. And then finally was the lemma analysis, which was the R package we used to generate a design matrix and then form a linear model. So now this is just another slide showing why we use lemma. Basically, it allowed us to account for variations in the data quality, accommodate technical and biological heterogeneity, and reduce noise, like Yves was mentioning earlier. And here is just a little graphic on kind of the criteria we were using for the differentially expressed gene discovery. So there were two kind of buckets. The first was statistical significance, which included the p-value and false discovery rate. And the second was the effect size, which was kind of the scale, and that included the fold change and the signal to noise ratio. Now we're going to be moving on to functional analysis, but first we'd like to share a little bit about how important team structure was as the project became more complex.
Within each section of the pipeline, our two larger teams are split into smaller 2 to 4 person sub-teams, where they apply different tools with respect to each step for the purpose of comparing and contrasting the output among groups. In the quality control step, we discussed teams use the AFI package, while others use a simple AFI package. And in functional analysis, sub-teams worked on different tools as well. The functional analysis part was fairly different between the two sessions, and we'll start off by looking at the June session.
Once we have identified the top dysregulated genes through differential analysis, we can give them a more applicable biological meaning through functional analysis. In this step, we identify the most common cellular processes and functions related to our dysregulated genes and identify the roles of proteins these genes code for. Throughout the course of the pipeline, we use many different tools for functional analysis. The first of these is gene ontology analysis, using the enrich-GO function in R. Gene ontology uses a comprehensive model of biological systems to identify the top biological processes, cellular components, and molecular functions associated with dysregulated genes. It is often useful to compare differences between overexpressed and underexpressed genes, because it is the imbalance between the roles of up and down regulated genes that leads to the progression of disease. For example, in the case of colorectal cancer, which we studied during the internship and is shown in the bar graphs, we see that many upregulated genes are involved in RNA metabolism and catabolism, while many downregulated genes are involved in signaling pathways. This makes sense because in the cell cycle, RNA primers are built and broken down during DNA replication, and intercellular signaling decreases. And these different processes help explain the continual replication of tumor cells. Another useful tool we explored was KEGG analysis. KEGG stands for Kyoto Encyclopedia of Genes and Genomes, which is a collection of manually drawn pathway maps representing molecular interaction networks for a variety of cellular processes. Identifying the most significant pathways of dysregulated genes helps us understand the mechanisms of a disease. During our analysis, we use the enriched KEGG function in R to compare our list of genes to the KEGG database and identify the most common pathways of the dysregulated genes involved in colorectal cancer. When looking at the dot plots, we see that upregulated genes are involved in RNA transport, the cell cycle, and DNA repair mechanisms, indicating that DNA replication has increased along with cellular division. Downregulated genes are involved in many signaling pathways, similar to what we saw with gene ontology analysis. Of particular interest are the RAS and MAPK pathways, which control cellular processes including inflammation and cell division and have been linked to cancer. In addition to functions in R, we also used external web-based tools to perform functional analysis. The EnrichR website analyzes a list of gene symbols and outputs enrichment data of common annotated biological features, including gene ontologies, wiki pathways, and KEGG pathways. David, the database for annotation, visualization, and integrated discovery, analyzes a list of gene IDs and outputs the enriched biological themes, including gene ontology terms, KEGG pathways, and interacting proteins. Finally, Metascape, a web-based gene annotation and analysis resource, analyzes a list of gene symbols and outputs quality control analysis, gene annotation, and enrichment analysis of pathways, processes, and protein-protein interactions. These tools are useful for performing many types of analyses at once. All of these tools are similar, but differences in website format, output contents, and output format may make one more useful to a particular analysis project. Our interns found these external tools very exciting, and we would love to hear your thoughts and recommendations on using these for further study. It is very interesting to study functional analysis, and it is important to know how to interpret the results. Identifying and studying enriched pathways gives us a biological context for dysregulated genes and shows us their importance to certain cellular processes. This also helps us to understand the mechanisms of disease. Additionally, identifying the most disease-relevant and involved pathways among the enriched pathways helps us find targets for drug discovery. By developing drugs that regulate the relevant effective pathways in a disease, researchers can create more effective treatments. One of the issues we faced during the June session was that our technical focus took away from the big picture, making it difficult to understand the specific roles of dysregulated genes. We addressed this problem in the July session by using a wider range of tools, which Anya will now explain.
Thank you, Alicia. Following the June session, we wanted to bring more of the biological aspects of bioinformatics to the forefront. And the first tool we used was the string database. The string database contains known and predicted protein-protein interactions, or PPIs, including direct and indirect associations. Given a list of differentially expressed genes, the web application for this database constructs a network of genes and their PPIs with other genes. The primary purpose of using the string database was to identify targetable genes based on their interactions. We can identify these genes by comparing the number of connections each gene has On the plot, those with a greater number of connections are key drivers. So for example, on this network, we have 2 clusters of genes with an average connection count of 6, and then 1 gene in the middle called MYC, or MYC, with 20 connections. So in this case, MYC would be considered a key driver. MYC is a proto-oncogene that codes a protein involved in cell cycle progression, apoptosis, and cellular transformation, which are all processes that are known to be abnormal in cancer. Following the identification of key drivers, we looked at global gene set enrichment analysis, or GC. This analysis, unlike gene ontology, looks at enrichment across all the genes in the pathway. It doesn't require cutoff values, and so is considered unsupervised, and allows us to investigate the additive effects at a systems biology level. The goal of using this analysis was to discover specific gene sets defined a priori that show statistically significant differences between 2 phenotypes, in our case, cancer and normal tissue. For our analysis, we used the hallmark gene sets from the MSIG database, which represent well-defined biological states and processes such as DNA repair and cell cycle regulation, shown in red and green respectively on this figure to the right. The GC function outputs 3 plots on top of each other. The top plot plots the enrichment score of the gene set at each position in the gene list, which is ranked by log fold change from the DGE analysis. The middle shows where the members of the gene set appear in the gene list, and the bottom shows how a gene correlates with the phenotype. Gene sets that produce a peak at the beginning or end of the plot, or the left or right, are the most interesting to look at since they indicate that the gene set is particularly enriched in either up or down regulated genes. In the figure shown, we are looking at gene sets from DNA repair, cell cycle regulation, and MYC targets, which all have a peak and are related to abnormalities in cancer cells. To investigate specific genes further, we used another web application called GEPIA. GEPIA stands for Gene Expression Profiling Interactive Analysis and provides various tools for analyzing expression data of samples from the Cancer Genome Atlas, or TCGA, and the Genotype Tissue Expression Project, or GTEx. We used it specifically for survival analysis. We wanted to identify differences in clinical and prognostic importance of identified targetable genes, or candidate genes. The figure shows the survival analysis for FOXQ1, a gene involved in embryonic development, cell cycle regulation, tissue-specific gene expression, cell signaling, and tumorigenesis. This gene was the top differentially expressed gene from the DGE analysis, and you can clearly see a difference between the survival rate of patients with high transcripts per million, or TPM, and low TPM. Those with higher levels have a lower survival rate, indicating that FOXQ1 might be a good gene to target to improve survival. For the MYC gene, which I've mentioned before, the difference wasn't nearly as pronounced. This is possibly due to the fact that it is the mutation, rearrangement, and translocation of MYC that is associated with tumors as well as its overexpression. So the TPM of MYC has a lesser effect on survival rate since it can promote cancer in other ways. Moving away from genes, we also looked at regulators using gene concept networks and the C3 gene sets from the MSIG database. If you recall the research paper at the beginning of this presentation, it concerns the construction and analysis of a competing endogenous RNA network. The network result from the paper included a long non-coding RNA, a microRNA, and a messenger RNA. And if you've noticed, none of our previous analysis concerned RNAs. So in order to include this aspect of the paper in our research, we conducted regulator analysis using the C3 gene sets to identify upstream master regulators related to our top differentially expressed genes. These gene sets contain potential targets of regulation, such as transcriptional factors, microRNAs, and long non-coding RNAs. In the gene concept network shown, the genes are connected to regulators that they are associated with. The regulators shown are all members of the EF family of transcription factors, which regulate genes essential for DNA replication and cell cycle progression. This analysis, along with the string database, GC, and GEPIA, allowed us in the July session to really get a more in-depth understanding of the biology behind the gene lists we were producing. In this presentation, we've identified 2 gene sets, MYC and FOXQ, that are related to pathways, gene sets, and regulators associated with cell cycle regulation, a process that is highly abnormal in cancer tissue. And so this process that we've learned could also be applied to other data sets, which it has been in our final projects. And we can identify genes, pathways, and regulators of novel importance. So that concludes our overview of the summer projects. Erin will now lead our panel discussion.
Very nice presentation.